So in Chapter 3, we're talking about adaptive immunity, and we need to look at the players. In Chapter 2, you saw a whole bunch of different players for the innate immunity side of things. And there were lots of different cells, lots of different molecules involved, and lots of different processes. And the innate immune response is really the second line of defense. What's the first line? Just to pull things out from memory. What's the first line of defense? All those barriers, all those chemical and mechanical mechanisms that we talked about previously, those are all the first line of defense. And then if those invaders get past those guys, then the cells, proteins, and other molecules that we talked about in the innate immune response start to kick in. And then if they can't deal with it, then we start to get an adaptive immune response. If you have a puncture wound, and it's an organism that you've never seen before, and a few cells get in, and it's not some pathogenic organism, then the innate immune response, those players are going to deal with those cells pretty quickly and take care of those guys and get rid of them from the area. But if you get somebody who can avoid those responses from the innate immune side, they may be able to survive some of those processes that you looked at and discussed in the innate immune response, and then adaptive immunity has to kick in. We've already talked about how it's slower, and part of the reason is because if you've never seen that antigen, you have to start making some of these molecules very specifically from the beginning when they're not in existence. And so the main players of adaptive immunity are two cell groups, really. But it's hard to break it up. Most textbooks compartmentalize it like this textbook tries to, but you have to realize those players in the innate immune response are coming back and interacting with the adaptive immune response. If you separate them in your mind, you won't understand how the adaptive immune response gets started in most cases. So you have to bring it together and homogenize it, build a picture, and usually I describe this class like a motorcycle. If you have enough money, you can go to the Harley-Davidson shop and you can buy a pretty nice motorcycle. But if you've ever bought one, you know it doesn't have a lot of chrome, it doesn't have a lot of bells and whistles, and you're going to want to make it your own. And so as you're going through this class, these 3 chapters are really that base model Harley-Davidson with nothing special on it. And then as we go through the subsequent chapters, we're going to begin to put some really important stuff on. But we're going to get some details of how this works. And this chapter, a lot of it should be reviewed from, like I said, micro and anatomy and physiology. Hopefully you've all had that. So the main players in the adaptive immune response are going to be B cells and T cells. And those B cells and T cells have specific receptors on their surface that we call the B cell receptor in the case of the B cell, and the T cell receptor in the case of the T cell. The B cell receptor also gets another name. Sometimes it's called surface immunoglobulin, and that's just because of the structure of these proteins. And you can see this kind of blocky structure of this, and it's got lots of different domains here, and actually this bottom part of the heavy chain is going to have multiple domains depending on which immunoglobulin we're looking at. But those immunoglobulins have very specific structures, and the reason they get called immunoglobulins is because there are lots of beta sheets in each one of those blocky areas. Do you guys remember from your cell biology, anatomy, physiology, micro course, what do beta sheets look like, or beta pleated sheets look like?
They're like when you fold a piece of paper, like an accordion.
Beautiful. So it kind of zigzags this way, and if you get enough of those beta sheets in a stretch in a polypeptide, they can fold around. And I'll do it this way since my fingers look kind of like beta sheets. They can fold around like this, and then we call them a structure that's called beta barrels. They form a barrel-like structure, and each one of those little domains that's kind of blocky there has that beta barrel structure down inside. And so along the way, if you haven't taken Dev Bio yet, you'll probably hear immunoglobulin-like structure, and that's what it means, that that particular protein has beta sheets that fold into a beta barrel, and thus it has that immunoglobulin-like structure. So when they're on the surface, we call them immunoglobulins. We can call them secreted immunoglobulins when they're released from the cell or secreted out of the cell, or we can call them antibodies. They mean the same thing. They refer to the same molecules. They're very, very similar, but there's some slight differences between surface immunoglobulins and secreted antibodies. And we'll come back and talk about it. But over here we've got a cell that's got this surface immunoglobulin, and if it sees its antigen and it binds to this antigen binding site that's right here, pretty soon it'll differentiate. It'll go through some developmental processes. It'll change down inside, and pretty soon it'll continue to put some on the surface, but pretty soon it'll start making immunoglobulins or antibodies that are going to be secreted out. And that's that differentiation process between a B cell and the plasma cell that we've talked about previously in the previous chapter. The T cell, let me go back and talk to you a little bit more about the structure. So immunoglobulins have 4 polypeptides. They've got 2 polypeptides that are called the heavy chains, and in both of these drawings, those are the blue polypeptides. And both of those polypeptides are going to be identical in amino acid sequence. They are blue, right? I see some of you squinting, but okay, good, because they could be purple and I wouldn't know it. So the blue ones are called the heavy chains. And then the red ones are called the light chains, and the reason they got those names is because the red ones have less amino acids. So in the old days, they would separate things out either on a density gradient, in this case they probably didn't use that, or on a SDS page gel, which hopefully you've heard about in other classes. And you can separate based on size, and the light chains have less amino acids, so they migrate faster, they're smaller, and the heavy chains have more amino acids, so they obviously have a higher molecular weight. You can see one important feature of the surface immunoglobulins. They're embedded into the cytoplasmic membrane of that B cell that has them on the surface, and they have a transmembrane tail that's important. One of you put a question about signaling processes in the innate immune response. That transmembrane tail on the immunoglobulins is also involved in signal transduction on B cells. And there's a few other things that the immunoglobulin does, but we'll talk about those. So you've got that transmembrane tail with the cytoplasmic domain that's down inside when it's on the surface. So 4 polypeptides, 2 genes, coded for at 2 distinct loci. What's the difference between a gene and a locus? Somebody remind me from your genetics class.
A locus is where a gene's located.
So a locus is the position on a given chromosome where a given gene is located. So in other words, there's 2 different genes. They're on 2 different loci there, or each one is on a different locus, and those actually reside on different chromosomes in humans. And so we'll come back and talk about that. A lot of the detail that I'm giving you now is actually in Chapter 4. We're going to come back and talk about how we get diversity and things like that in Chapter 4. Then there's the T cell receptor. So this guy's on the surface of the T cell, and it's only 2 polypeptide chains. Both of these polypeptide chains, so it's also a heterodimer. Remember that term that we used earlier when we were talking about the toll-like receptors? The T cell receptor is a heterodimer. One is called the alpha chain. It's coded for by one gene, and the other is called the beta chain. It's coded for by a second gene. Again, both of them at different loci on different chromosomes. And the important part is you get those polypeptides. Initially, they're stuck on the surface of the cells, but they've got these regions here and these regions here that are very specific for very specific antigens. They can only bind to a very, 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 very few antigens. Maybe 1,000th or a 10,000th or even less of the antigens that come into the body will be able to bind to one specific T cell. And if they bind, they may not bind real strongly. They may just bind enough to send some stimuli down inside of that cell, because those antigen binding sites are very, very specific. And we'll look at that a bit here in this chapter, but even more in the next chapter. So these are the molecules on the outside of these players, these 2 types of lymphocytes that are important in adaptive immunity. So ultimately, as a part of the adaptive immune response, at some point we're going to hopefully get antibodies produced to a given pathogen. And the point of this slide is just to show you that antibodies that are produced are very, very specific to the pathogen that starts that production process. So let me see if you guys, this is a quiz question. What's the definition of an antigen? What's the definition of an antigen? Antigen. It's actually an abbreviation. What's that?
Antigens?
Not quite. It's something that generates antibody production. Antigen generates antibody production. That's how it got its name. These molecules that can cause the system, an animal, human, whatever it is, to produce antibodies, we're going to call antigens. Okay, that's how it got its name. Any molecule that can stimulate an antibody response. antibody production response. Okay, so in this case, many of you might have been vaccinated, others of you had the measles along the way, and what they're trying to say is that you get a number of antibodies that are produced. In this case, you see two specific antibodies that are produced to specific antigens on the surface of the measles virus in this case. Could be any pathogen that you chose, but they chose the measles virus, and there's a couple specific antigens, and so you see some of them binding to the surface of the measles virus to this yellow triangular antigen. Others bind to the surface this blue semicircular antigen, and the ones that bind to the blue one can't bind to the yellow triangle, and vice versa. Okay, they're specific. Down the road, you're going to find out that a number of different antibodies might be able to bind to that yellow triangle, not just one, but a number of types. Some will bind more strongly than others, and we'll talk about some of the functions of those. Same thing here, but the point is, once you've been immunized against that measles virus, and then now you get the influenza virus over here, those antibodies that you got produced to the measles virus likely won't protect you from the influenza virus. In fact, to make it even more clear, Diane, I need your recording again. I hit stop in the middle. And she's given me 3 recordings. I'm going to put those up for you, but somehow I hit stop in the middle. So once those antigens come in, specific clones are going to grow, divide, differentiate, and begin the process of going down the way through their effector cells. And we'll come back and we'll talk about this next time. Don't forget